Good afternoon, my name is Brian Joseph and De La Cruz. This video is talk about methods of identification other than by fingerprinting. The first is anthropometry Bertillon system. Anthropometry, introduced by Alphonse Bertillon in 19th century and spread it to the world. In 1914, fingerprint was officially adopted in France, replacing the Bertillon system. Composition of Bertillon system. Number 1 is anthropometrical signalment, measurement of the body, head and limbs. Number 2 is descriptive signalment, clear and specific system of description of an individual. Number 3 is specular mark, signalment by indelible marks such as scars, tattoo, birthmarks, moles, and etc. Alphonse Bertillon, a clerk in the Prefecture of Police of Paris, France. System of classification, anthropometry, first method of classification, also known as the Bertillon system. This method laid groundwork acceptance of fingerprint as a scientific method. The second topic is portrait parley. Introduction. Portrait parley, these two French words mean speaking likeness. Portrait parley can be defined as a rigorous system for verbal description of physical characteristics of the subject. It is the base of the Bertillon method of criminal identification measurement of the human body. It is still practiced today to some extent. Following features considered in portrait parley: face, oblong, oval, round, rectangle, square, triangle, diamond. Widow peak. The example of the feature is the pointed of the hair. Skin color, very fair. Fair white skin, light brown, moderate brown, dark brown, deeply pigmented, dark brown to black. Hair types: straight, wavy, curly, coils, undefined coils. Eyes: round, almond, droopy, droopy hooded, Asian, round, Asian, etc. Eye color: gray, green, blue, honey brown, black. The color of eyebrow: black, dark red, dark brown, medium red, brown, light red, blondie, light blondie. And the types of eyebrow is thin, natural, thick. And the distance between eyes: wide set, downturned, almond eyes, close set eyes, hooded eyes, almond eyes. And the average distance between eyes, the pictures to example the distance between eyes. Bird style: Hollywoodian, Old Dutch, ducktail, Winfield pencil, Angkor, hand lever, Super Mario, Mouton chops, to name a few. And other types of chin. This picture is example of shape of chin. And next, types of nose. This picture is the shape of nose. And next, nose length, bridge to tip. Number 1 is 5, extra small. Number 2, 0.75, small. Number 3, medium. Number 4, 0.25, large. Number 5, 5, extra large. The forehead and cheek freckles is example is the mark of many mark of faces. And the next picture is ear: large, small ear, sticking in and sticking out. And the next picture is hairy ear. The example of the picture is to show the hairy ear. And the next picture is types of lips: closed points, flat tops, roller coaster, large, lower. Occupational marks. Some marks develop due to the kind of occupation performed by an individual. Some examples are given below. Tailor: marks of pin pricks. Dishwasher: hands are rough, sooty, and bigger in shape due to continuous exposure to water. Truck driver: corns on palm. The next topic is forensic odontology. Forensic odontology: proper collection, interpretation, evaluation, as well as presentation in court of dental evidence during the conduct of criminal and civil proceeding. Analyzing teeth. The first picture is permanent teeth and the second picture is baby teeth. The next picture is individual characteristic: the size of tooth, shape of root, placement of tooth, quality of teeth, combination of dental work done, crown extraction, bridge, fillings, root canals. Computer odontology, automatic dental code matching. Bots are run through the computer, find a match. Odonto search, compare the databases of missing people, felons, government workers. Automatic dental identification system. A few minutes will produce a list of people who have the same dental code number. 3D bite mark analysis. 3D scans of dental are used to generate overlays using various pressure and deviation. The overlays of compare will be the photograph of the bite marks. The next picture is teeth basic. Approximately 32 teeth in adult man. There are 4 types of teeth: molars, premolars, canine, incisor. Teeth differ in size, shape, root type. This picture is teeth through the ears. The start is baby tooth to the upper and the lower. The next picture is bite marks. This picture example of bite marks. And the other picture is analyzing bite marks. Bite marks are photographed with a scale. Bite marks on skin are taken over repeated intervals. Cast of impression of taken. Impression traced into transparencies. Cast of suspect teeth are taken. Comparison between suspect cast and bite mark. The next topic is forensic anthropology. Forensic anthropology is a special subfield of physical anthropology, the study of human remains that involves applying skeletal analysis and techniques in archaeology to solving criminal cases. The next picture is determination of sex. Cranium. Crest and ridge more pronounced in males. Chin significantly more square in males. Forehead slope more in males. Mastoid process wide and robust in males. The next picture is determination of age from bone, sign of wearing, and antemortem injury. The first picture is occupational stress, wears bone at joints. The second picture is surgeries of healed wound, aid in identification. Determination of race Caucasian. The picture is orbital opening rectangular or square. The nasal spine very small. The next picture is nasal ceiling or guttering. Feel the base of the nasal cavity on either side of the nasal spine. Caucasian sharp ceiling, mongoloid rounded, negroid no ridge, guttering. The next picture is the bone we're interested in. Example of the picture: skull, humerus, pelvis, femur, tibia. The next picture is determination of sex. Number 1 picture is male, number 2 picture female. The subpubic angle is much wider in female than into male, typically more than 90 degrees and less than 90 degrees respectively. The next picture is determination of stature. Long bone length, femur, tibia, humerus is proportional to height. There are tables that forensic anthropologists use, but this also depends to some extent on race. Since this is intact, there are confidence intervals assigned to each calculation. For example, imagine from a skull and pelvis, you determine the individual was an adult Caucasian. The height will be determined by humerus length 30.8 cm. The next topic is forensic serology. Forensic serology is the application of the study of blood, semen, saliva, and other body fluids to legal matters. The field generally is comprised of the detection of enzymes and antigens as the identification of seminal stain of blood typing, ABO and secretory status, and DNA typing. The composition of blood. The next picture is forensic characterization of blood stain. Number 1 is luminol, number 2 Leukocam, green, number 3 Castelmeyer. The luminol is red blood cell contains hemoglobin, the protein responsible for transporting oxygen. Each HB contained for iron contain hemes. Luminol react with iron. The next picture is blood typing. Blood typing is human serum containing specific antibodies can be purchased. Separately mix a drop unknown blood sample with drop of each anti-serum. The next picture is blood stain analysis. Categories of blood stain: passive dripping, transfer smearing, projected. The next picture is projected blood stain analysis. There are two important determinations: direction of spatter, angle of impact with surface. The next picture is forensic characterization of saliva. Saliva is a mixture of many components, 99% water, mucin proteins help in swallowing, amylase enzyme to help digest carbohydrate, buccal cells, good source for DNA. A simple test for saliva involves mixing starch, iodine, and a sample for the presumed saliva together. Starch as iodine are a deep blue color when mixed together. The amylase break down the starch, however, and the color fades. Take about 15 minutes at 37 Celsius. The next topic is DNA fingerprinting. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene is compared with a DNA sample from a suspect and the two DNA profiles are a match. Then the evidence came from that suspect. The next picture is fingerprints and DNA strand. Fingerprinting relies on the unique pattern made by a series of DNA fragments. After separating them according to length by gel electrophoresis and the DNA strand, samples from different suspects, the victim, and samples from the crime scene are the first verified. The samples are then proceed to generate a set of DNA fragments.